I'm going to introduce the chair of the next session, which is the panel discussion on emerging trends and technological advances. So the chair of the session and a speaker as well is Dr. Gwenn Hansen, which is the Chief Scientific Officer at Nurix Therapeutics, a company focused on discovering and developing next-generation targeted protein modulation therapies. Since joining Nurix, Gwenn has focused on developing a robust pipeline for early-stage discovery by developing a platform based on DNA-encoded library technology and leveraging it across a range of target classes, particularly E3 ubiquitin ligases. Thank you, and welcome, Gwenn. I don't see Gwenn. Gwenn, are you here? I see the slide being shared.
Yes, she's here.
I believe, Christopher, you are sharing the slides, correct?
Gwenn's sharing them.
Oh, Gwenn's here. Okay.
Yeah.
So we can't hear or see her.
Okay, sorry, I was
there you are.
finding the link. So can you confirm that my slide presentation is in slideshow mode?
It's not. We see the presentation, but it's not in slideshow mode.
Okay.
It's in slideshow mode now.
Great. Yeah, absolutely great. So thank you, Sareen, and I want to thank everybody from NCATS as well as on the organizing committee for putting together this really great webinar. I think we've covered so many topics, and we really have talked about the current state of DEL in a way that makes this end session even more difficult, because I think we've talked about so many of the emerging challenges and applications and utilities of this technology that it's kind of hard to cap it off, I think, appropriately. But we have prepared a panel discussion for this. It's going to start with 3 short talks. I'm going to give the first one, followed by Brian Pagel, Jeff Messer, and Ryan Potts. And we're focusing on those avenues, not so much in the primary execution of the primary discovery technology itself, but where it's being utilized and where it's going to get more utility out of the platform than simply the great value of simply finding and enabling inhibitors for targets of relevance in disease. So I'm going to jump into my talk first, and Sareen has already given the title, and there's a disclaimer. Obviously, everything is forward-looking because I'm really just talking about where Nurix is going. So let me set the stage by saying that what I'm trying to accomplish here by giving you a little flavor of the Nurix platform is more to tell you how we think about the value of this technology as a cornerstone of a small biotech company. And it kind of gives you an illustration, I think, of how valuable it can be when applied in very specific ways. So to start off that theme, what I want to characterize is this whole idea of targeted protein modulation, and that's the way that we think about it at Nurix. And we really see this as a new therapeutic modality that really changes the way that most of us think about being able to use small molecules in the cell. So the history of drug discovery has come from inhibiting individual proteins to developing protein therapeutics in terms of antibodies and peptides. And most recently, there really has been some good forays into nucleic acid-based therapies. All of these approaches kind of have advantages and disadvantages, but the thing that really is drawing the attention of the field of small molecule discovery right now is the ability to really affect very dramatic changes in individual protein levels by taking advantage of an inherent function that exists in the cell. It's the ubiquitin proteasome system, which is present and necessary basically to regulate, eliminate, or modulate proteins and their function, and that protein modulation can be far more rapid and complete than transcription alone can be. And so by taking advantage of this particular modality that the cell already has through small molecules, we really think that we can achieve some therapeutics that haven't been possible before with simple inhibition or protein resources. So the way that Nurix set up its platform leveraged that effect, and it did so primarily because we really were founded to study E3 ligases, and they're one of the largest classes of proteins in the human genome, more than 600 certainly, but they are really very poorly studied and never thought to be druggable targets. And so many people now are approaching these difficult to drug targets in different ways, and we did as well. And what we decided in the very beginning is that we needed a resource that was able to interact with the protein surface, not limited by its ability to be read out in a biochemical assay. So that's one of the very strong features of the DNA-encoded library technology. In that, I mean, actually, in the beginning of the field, it was thought to be a liability in that it couldn't focus in on just the most relevant thing if you were looking for biochemical functionality, but it truly is an advantage when you're dealing with difficult to drug targets and you can take a ligand and do more things with it. So we centered our platform on DNA-encoded library technology. We really focused on E3 ligases, but also on the identification of multiple different binding modes from those targets of interest. And the thing that we've heard a little bit about, and we'll hear more later in this panel, is that not only do you get a wealth of data of highly related compounds because of the way the libraries are designed through split and pool combinatorial chemistry, but by applying very complicated but interesting analyses of these very complex data sets, you could even read between the lines of the compounds you can't make in your library to potentially have a greater reach than what is just synthetically possible. So to start this out, because I'm focusing on targeted protein degradation or modulation, I want to really kind of set the stage to say, why is it that everybody's interested in this? So a few of the reasons are here: the ability to remove a faulty or disease-causing protein completely from the cell, and that really is the most appealing as you look at the data that comes out of these studies. Treating resistance mutations by being able to touch on a protein surface and not have to fully occupy it and avoid mutations that would reduce that ability to occupy the protein is another advantage. Going beyond stoichiometric inhibition, not needing as much drug on board to accomplish that goal, and to address difficult to drug protein targets, of which ligases certainly are part of it. So I do apologize for the simplicity of this cartoon, but I want to make sure, because this isn't a TBD conference, that everybody understands what we're talking about. E3 ligases are part of a cascade of the ubiquitin proteasome system, and we talk about them because they're the terminal step of that cascade that really recognizes the protein substrate. And by doing so here in this cartoon, what E3 ligases are able to do is cause a transfer of ubiquitin, a protein tag, in a very specific fashion. And if that ubiquitin tag is formed properly with the right linkages, it will trigger shuttling to the proteasome and degradation of the protein. Now, in the context of taking advantage of this system that exists in the cell, if you create a PROTAC, or what we call a chimeric targeting molecule that can touch on your target and also recruit a ligase, you can make the ligase transfer ubiquitin to your chosen protein of interest, and that results in the degradation of that protein. The reason any of this is really relevant for this conference is that I think early on in this process, when we were really focused on E3 ligases, we became very enamored with the idea that DEL might actually be the ideal technology for enabling this this mechanism of protein degradation to be expanded. DEL obviously identifies compounds that bind to proteins, and binders are what drives this process. And it also affords the access to the very largest collections of compounds that can be used for that type of affinity screening. So these weren't necessarily seen as advantages of this technology in the early stages. Having a lot of compounds obviously is always good, but if you can't focus them in on just what you want, it's a problem. But for targeted protein degradation, that is absolutely what you want. And the more binding modes or the more binders you have, the better likelihood of achieving your final end goal of a very effective degrader. That the third point here, I think I've already mentioned, it's almost better if your binders don't modulate the protein target in affecting degradation. And that is because you can avoid some off effects if your binder would modulate your target and could modulate another target if it was there at enough concentration. So seemingly all of these things seem to align to really focus the DEL technology toward this particular aspect. And one final point I think we've already heard from the previous speakers is the chemical linker to attach to DNA now also is not a liability, and that it really does guide your early degrader design. So illustrated in this graphic is really just what I said. You can think of screening of both your target of interest to find a chemical binder, we call that a hook, or you can think of an E3 ligase as a target of interest to find a binder, and that we usually call a harness. And the linker that attaches to DNA is really pointing out, as we've heard, towards solvent, and by joining those together, you can really create almost a Lego-like thought process of linking your harness to your hook with a linker to form that chimeric targeting molecule in our vernacular. And there are many other things that I'll talk about later in the talk. I want to get to them, and Paul has shown this slide. But the whole idea of being able to shuttle in many different ligases and many different targets simultaneously, and really search through that data to find all the binders to put into your combinatorial process to build molecules that you test for terminate complex formation or cellular degradation, really gives this technology a boost from the empirical base that most of us are operating in. So I have just a few more minor data slides where I want to make some key points of emphasis. One of them is that when you screen through DEL, you do find a mixture of binders because you're really not focusing on any one particular biochemical readout. And so by doing that, in this illustration, one of our early ligase screens, we were able to find binders that sit on the front side and the back side of the protein. And the reason that this became very important to us is that to use a ligase as a degrader or to degrade a ligase or another target, sometimes you have to try multiple sites of a protein, not only multiple vectors out from your binding element. And so this is really an advantage of this technology. And so the more series and distinct binding modes of proteins that you can follow up in this technology, the better off you are in terms of affecting your goal. So two other little bits of topic I want to share is that the other advantage of this technology, as we've heard many times, is that you can run many parallel screens. And some of those parallel screens could be run for highly related protein members. In this case, I'm showing a paralog screen that's giving you a readout of two different targets that are relatively similar by looking for things that are highly enriched for target A as opposed to things that could hit both targets. What you can find is either a selective hit, but what you can also find is a selective binder and degrader. And so in crystallizing some of the top hits from that particular screen and looking at the DNA attachment point versus that, and the non-selective hit. So on the left, you're seeing the DEL molecule in blue and a non-selective molecule in yellow. These sat in our, this is a kinase target, these sat in our pocket in a very different place, not that unsurprisingly. The selective molecule sat out toward the surface of the ATP pocket. The DNA attachment point was out farther away from that protein surface. But when we overlaid it with a published optimized hit that came out of somebody's shop in the literature and really looked at the location of the DNA attachment point of our DEL hit straight from the library, what we found is that the DNA attachment point and the linker attachment point that was chosen in that optimization campaign were very similarly located. That was not true, obviously, of the non-selective hit. So there are some things that you can read out of the DEL hits, but you do also need to add that additional layer of thinking about what are you trying to achieve with your primary hit before you go forward into your campaign. But what was also very exciting to us, very early data from our work here, is that taking that selective hit into a very simple few compound design, linking it up to a ligase binder, and putting it into a cell-based assay, we were able to achieve a very potent, with a DC50 of 6 nanomolar, and very selective kinase A versus kinase B degradation with really no optimization. Now, obviously, not every campaign works this way, but it is really possible to get out not only highly potent, but highly cell active and effective degrader compounds straight from a Dell screen. And that advantage for a small company or a small lab is really hard to measure in terms of your ability to operate with success. So just a few slides to drive home the point that targeted protein degradation is really on its way into the clinic. This is our BTK program, our lead program that we chose really to learn about the field. After a bit of optimization, this is BTK CTM1 labeled because it was early in our process, certainly not the first CTM that we made, but we were really excited to see, as we had optimized in cell-based systems, that we were able to get very effective removal of protein in patient samples, particularly CLL patient samples. And this degradation correlated with highly selective degradation of our target protein BTK, as opposed to any other protein that we could measure by proteomics. And looking for those aspects that I mentioned before, that differentiation of a mechanism of degrader versus inhibitor, although in comparison to many of the top agents for treating lymphomas and CLL driven by BTK, we didn't have a really striking differentiation of our degrader, and again, an early degrader here, BTK CTM2, versus say ibrutinib or acalabrutinib. But what we really did notice is when we went into resistant patient samples, so this is an ibrutinib resistant lymphoma cell line, we could really see the difference between removal of protein versus inhibition, or covalent inhibition is in the case of ibrutinib. So taking this into animal models obviously takes more effort. Optimizing very large molecules to achieve oral bioavailability is a challenge, but it's something that we've been able to do many times now in our platform. And here I'm just showing data from tumor models, both were wild-type lymphoma on the left and ibrutinib-resistant lymphoma on the right. And we're able to very clearly maintain that differentiation of our degraders versus ibrutinib, which is the standard of care. And that oral bioavailability has, in our hands now, many times been able to translate into dosing of higher species, in this case, non-human primates. And so within a few hours, you can see complete removal of protein. And after many days of dosing, you have very effective maintenance of almost 10% to no protein with very little dosing of the compound. So we are entering the clinic with this degrader. We have a second degrader against BTK as well. And I think you'll see the result of this in the next several years, as there are many, many degraders, I think, now in the clinic or entering the clinic in the near term. And so this will, I think, read out soon to see how effective it will be at really making a dent in drug discovery. So just to conclude, I would say that DEL is fit for purpose. It enables discovery for proteins that we really thought were previously undruggable, and it has a much lower barrier of entry than many other discovery platforms. So I'm sure I'm way over time. I couldn't check the chat. So I'm going to stop sharing now.